So my name's Mike Anderson, and I'm the general manager of our bioanalytical services for ICON in Europe. And I wanted to talk to you guys about something a little bit different today, and it's more about what's the body's response to monoclonal antibodies upon exposure. And more specifically, what's the immune system's response? So to follow up on Michael's first talk of the day, when he showed the slide of the evolution of antibodies, early on in antibody development, there used to be quite a significant amount of mouse sequence in monoclonals. And some of the early antibodies, still more popular antibodies like Remicade, have a significant amount of mouse sequence in them, and they tend to stop working over time. The reason they stop working is your body's immune system is responding to those quite vigorously in some cases, and it creates a neutralizing antibody which doesn't allow the drug to work anymore. So immunogenicity assays and immunogenicity assay design are a fairly important facet of monoclonal antibody drug development. And it's something that really needs to be looked at quite early on. So this leads us into what really is immunogenicity and what do the regulators expect when you talk about immunogenicity. It can be really broken down into two branches. We have unwanted immunogenicity, which is really what we're going to focus on here today. And that's the formation of anti-drug antibodies against a therapeutic protein. There's also a branch of wanted immunogenicity, for example, in response to a vaccine, but that's not within our remit today. So unwanted immunogenicity is a major safety concern for biological drugs or biotech drugs. Now, what does major safety concern mean? Because it can mean different things to different people. So the regulators typically recommend the use of a risk-based approach when designing an immunogenicity program. And Maureen talked about this a bit in her talk. So what's the likelihood of ADA development versus the consequence of the development? And ultimately, what is the risk of your molecule? So thankfully, or maybe not thankfully for people, the risk of monoclonal antibodies is typically fairly low. If you get an immune response to a monoclonal antibody, there's not a huge risk that it's going to cross-react with an important endogenous protein. So we know that detection and characterization of anti-drug antibody is a key component of all large molecule drug development strategies. But what are the concerns that we have? So there's safety concerns. Anti-drug antibodies certainly can cause hypersensitivity reactions. So you can have infusion reactions upon dosing the first time you see a drug. Anti-drug antibody can neutralize the activity of an endogenous protein. So Maureen also touched on this with the EPO situation that was encountered. So there was changes in the way EPO was made. Patients were dosed with it. They made antibodies to the EPO they were dosed with that cross-reacted with their endogenous EPO, and a bunch of patients developed pure red cell aplasia, so they could no longer make red blood cells. That was really the spawn of the immunogenicity era, and it became a really highly visible in the regulators' eyes. It can change the efficacy of your drug, and this is evidenced by some of the early mouse-human chimeric drugs like Remicade, where they stop working over time. It can alter your PK. So not only can it cause faster clearance, it can sometimes extend the half-life of your drug. And hopefully, it causes no ill or adverse effects. So we know we have to test for ADA. We know we have to characterize ADA. What are the most common ways that we do that? And these are some terms you'll hear me reference later in the talk, but we have bridging immunoassays. So we have a bridging ELISA, a bridging ECL. Typically, what these are is you coat your drug on a plate, and then you have another variation of your drug that has a detector on it that can emit signal. So the blue antibody, and my antibody diagrams aren't nearly as nice as everybody else's today. The blue represents your drug, the green represents your anti-drug antibody. So in these cases, your anti-drug antibody bridge the two, hence a bridging assay. There's also a direct ELISA, or direct assay format, which we'll talk about very briefly, but it's more where you coat your drug on your plate, you add your sample with your anti-drug antibody, and then you have a generic anti-human IgG-AM detector that detects the ADA. There's pros and cons to all of these. There's no perfect assay to detect anti-drug antibodies. Typically, the bridging assays are good because they detect all isotypes. They're also highly sensitive to soluble target interference, which I'll talk about in a little bit. The direct assays are typically much better for non-antibody drugs because you can't coat your plate with a full monoclonal antibody because then your detector is going to see the drug. So pros and cons to all of them. None of them are perfect, but we do what we can. So immunogenicity assay design considerations. When you're kicking off a drug development program, you really need to think about how you're going to set up your immunogenicity assay. What are the pros and cons of each of those assay formats? And there's a lot of questions that you need to be able to answer and talk through as you go through. So what's the drug structure? Well, for monoclonals, that's pretty straightforward. What's the mechanism of action or the target of your drug? That plays a role in both your risk assessment and future characterization assays like neutralizing antibody assays that you're going to have to do. What is the potential for solid ligand interference? So we're doing a lot of monoclonal antibodies that target inflammatory cytokines. If they're soluble in the blood and they can, for some reason, dimerize, instead of this ADA causing the bridge, you can have a dimerized target causing the bridge. And this has caused myself personally all sorts of issues, which I'll tell you about in a minute. What's the potential for pre-existing positives? So we do a lot of work in inflammatory autoimmune diseases like rheumatoid arthritis, where you have rheumatoid factor in the serum. Rheumatoid factor is an antibody that binds to antibodies. So our assays, by definition, will pick that up as an anti-drug antibody. What are your dosing levels and sampling time points? The question those are going to answer is how much endogenous or how much drug is going to be left in the sample when you're trying to detect the anti-drug antibody. What are your potential assay formats and how can they help mitigate all the things we just talked about? And ultimately, what is the risk of your molecule? So two of the most important considerations, in my opinion, are drug interference or interfering serum protein and the potential for soluble ligand interference. So I just wanted to talk briefly through those two. So the challenge that we have, and this is outlined by industry white papers and some guidances, what the FDA guidance going on 4 years being a draft. We have to develop a method with less than 500 nanograms per mil of sensitivity when anti-drug antibody is in the sample at microgram per mil or sometimes mg per mil levels. So we get a clinical sample in that has 50 micrograms per mil of drug, and we have to ensure that we can detect 500 nanograms per mil or less of anti-drug antibody in that sample. So you can see the challenges there. The ratio's skewed not in our favor. So it's something you need to consider very early on, and that's where your dosing levels and sampling time points come in. That you need to ensure that you're picking your immunogenicity samples that are drawn from the patients when there's the lowest level of drug possible, so you can minimize that interference. On the assay side, there's also a lot of things we can do to help improve drug tolerance. We can introduce an acid dissociation step. So if you lower the pH of the sample enough, it'll break those anti-drug antibody drug interactions. And then we can kind of reset the balance so our labeled drugs in our assay system have equal chance to compete. There's sample cleanup steps we can do. PEG precipitation can pellet higher molecular weight proteins, and you can remove a lot of the excess smaller molecular weight proteins. There's a lot of new published literature on affinity capture elution methods, which can remove excess drug. There's a lot of new technology coming out with spin columns, which will bind your drug and take away all the excess drug. You can purify out your anti-drug antibodies that way. It's becoming quite complex, so it's better to look at this stuff early on in your drug development process. One of the most important things you need to look at, especially for monoclonal antibodies, is can you have soluble target interference? So we've just encountered a situation where we had a bridging assay where the soluble target formed a multimer. Now we knew this going in, so we looked at it very early on in the method development process for the immunogenicity assay. We found that the target interferes at 1 nanogram per mil. And by interferes, I mean it bridged the biotin and sulfotag labeled drugs, and it actually caused a false positive in our assay. And through interactions with our client, we knew we could expect up to 100 nanograms per mil in the sample. So the proposed solution, which sounded easy at the time, was let's source an anti-target antibody. There were a lot of them available commercially, and see if it could just block that interference. I think we were through our 14th antibody that we sourced before we actually found one that worked. And we had to add it at 50 micrograms per mil, which is quite a high level. But ultimately, through that due diligence, we found an antibody, finally. And when we added it at a high enough level, we could get our soluble target tolerance up to an acceptable level. Now, why I laughed when I said soluble target interference has burned me personally, we had a situation about 2 years ago where we did this exact same paradigm in drug development. We thought we were great. The FDA had asked this particular client to develop a more sensitive and more drug-tolerant immunogenicity assay before they started their phase 3 trial. So they switched from a direct ELISA to a bridging MSD electrochemiluminescent assay. We did all this testing and development and validation. And it turns out when they dosed their drug, the soluble target took on the half-life of the drug and accumulated to about 30 to 40-fold more than we thought it was going to. And even though it was bound to the drug, it still dissociated and interfered with our assay. So we ran the first phase 3 trial, and we had 55% of the patients positive for ADA, for a monoclonal antibody. So needless to say, the client wasn't happy. We had to go back to development, develop a new assay right in the middle of a trial, and start over. So these all are reasons why this stuff needs to be looked at with great due diligence early on, because it can prevent a lot of headaches, time and money later on in the drug development process. So I wanted to branch out a little bit and talk about cut point assessments and ultimately how do they play into making sure your immunogenicity assays are set up appropriately from the beginning, and really focused on how do you deal with pre-existing positives. So we see a lot of people going into the RA field, inflammatory autoimmune disease field. So this is becoming more and more prevalent. So in brief, a screening cut point is the level of response of the assay at or above which a sample is determined to be a potential positive, and below which a negative. So it's a fairly important defining parameter in dictating which way your samples are going to go. Are they going to be called positive, or are they going to be called negative? So it's determined statistically from background binding from drug-naive samples. And binding may be nonspecific. It could be just assay background or matrix components, or it could be specific. It could be pre-existing ADA, as we see quite a lot in rheumatoid arthritis patients. So how do we set a screening cut point? We typically buy 50 or more drug-naive human matrix, typically from a commercial vendor. The preferred approach is to do a parametric assessment, where we run these over a certain amount of days. We do some very basic statistics, and we get a cut point, which we can say with a reasonable amount of certainty, if we run these samples, 95% of them are going to be below, 5% will be above. Because per the regulatory expectations, we're expected to target a 5% false positive rate. This assumes normality. And a lot of times when you get into populations that appear to have pre-existing ADA, getting a normal distribution is next to, if not impossible. So that begs the question, should we start to exclude outliers? Now, samples sometimes display high signal in these inflammatory immune populations, and it leads to the non-normal distribution. So some questions we have to ask ourselves as we go through, are these biological outliers? So is it something in the sample that's actually causing a high signal that we can tease out what it is? Or are they analytical outliers? Was it an analyst error? Is it something wrong with our assay? Is our assay not robust enough? How many outliers is too many? So if we run 50 and we have 10 samples identified as outliers, now that's 20% of your population. And you always have to think, this needs to apply to patient data. So are we going to see the same thing when we get into patients? Does that mean we're going to have 20% of our pre-dose samples coming up positive? Is there a propensity to have pre-existing anti-drug antibodies? So do we think, is there any chance that these serums that we're using to set a cut point have pre-existing ADA? We know we're supposed to target a 5% false positive rate, but is that always the best approach? Can we sometimes target a 10%? Is 15% too much if we're taking a conservative approach? And we can ultimately exclude those through our confirmatory testing if we happen to go a little bit higher. So it sometimes gets very complicated to figure out how to set a proper screening cut point. So if you have a situation like this, which is more typical in a rheumatoid arthritis population, it gets fairly straightforward. You have your defined population, from a signal, 150 to 300 RLUs. And you get a nice little outlier population, very bimodal, very easy to say, okay, these guys look like true pre-existing anti-drug antibody. Let's take them out, we get a normal distribution, we'll set our cut point with this population, and we'll deal with these guys in the data handling portion of the study. But what happens if you have a situation like this, where you have your peak, and then you just have a tail? No bimodal distribution, no two distinct populations. Do we force normality? So we have some outliers here. We can take them out, reassess it. We'll have a couple more outliers here. We can force that population in, but is that being fair to the actual population? Because this is going to, in theory, this is going to happen again in your study. So do these outliers really contain ADA, or is that just an artifact of the assay? Is that the normal variability? And ultimately, what we have to take into account is reporting of patient results. So if we leave all these samples in and set the cut point somewhere out here around 600, if we have a sample around 200 or 150, it's going to have to triple or quadruple in signal before it's going to become a positive. Is that going to be acceptable to the regulators? Probably not. They're probably going to say that's too much variability. Typically what we do is split the difference and exclude the first bunch of outliers, leave the rest in. If the data is not normal, we can do a non-parametric cut point assessment. But really, ultimately what needs to come out of this is we need to tease out the prevalence of anti-drug antibody versus the induction of anti-drug antibody. So the prevalence of ADA can be high, and it can have nothing to do with the drug you're giving the patient. They just have anti-drug antibody. But what we need to know is the anti-drug antibody increasing or forming based on exposure to the drug? And if we suspect that we have pre-existing ADAs, or we know we have pre-existing ADAs, can we develop around it within the initial stages of method development? Sometimes yes, but the majority of the time no, we can't really do that. So what is our other option? Our other option is to handle this during data reporting. And there are a couple of unique ways we can do that. So for the cut point assessment, say if we take a population like this, we try to drive the cut point as low as we can so we can ensure we're going to get positives in the majority of the population. We need a way to handle these guys, which we know are going to come up positive. So a couple of very straightforward ways excuse me, to do that are set a within-patient cut point. So we can look at ratios of pre-dose to post-dose within the same patient. And that's going to track increases over dosing time within each patient. So we can really tease out prevalence versus induction. Another way that's becoming more and more popular is we do it via a characterization assay, which we call endpoint titer assay. So we'll drive the cut point pretty low. We'll screen and confirm all the samples that we think are positive. And then we'll do a characterization testing, which is how many dilutions does it take for that sample to turn negative. And we can statistically track that patient before they were dosed to all their post-dose and see if they've significantly increased over time. And a lot of our clients have taken to this approach to, even if the patient is a pre-dose positive and a post-dose positive, if it doesn't increase over time, they'll ultimately call that patient drug-induced ADA negative. So I'll summarize now because it's almost lunchtime. But we know that careful antibody monitoring is necessary throughout clinical development. And so the point I wanted to get across is that well-thought-out assay designs early in the program can really prevent problematic issues later on in the development cycle. It can save you a lot of time, a lot of money, and a lot of regulatory questions. And with that, are there any questions?